biological molecules. They are precursors. They are the building blocks for the other biological molecules like proteins, other keto acids and other molecules, nitrogen-containing compounds, biological compounds. Because amino acids are containing nitrogen molecule, rich source for the nitrogen molecule. So wherever nitrogen is required, amino acids they can get. Okay, so they are precursors for other biological molecules. So here you can see the how proteins can be built up by joining one amino acid to another amino acid upon using water molecule and upon creating a peptide bond. Here you can see one amino acid or one another amino acid or two. So when they join, water molecule will be removed and peptide bond will be formed C double bond NH. This is the peptide bond of two amino acids. So in a protein, several peptide bonds will be there because all amino acids join together by peptide bond in a protein. Okay. So now you can see energy source here upon catabolism of amino acids. So it provides ATP synthesis. It provides high energy compound synthesis here. So ATP is synthesized and waste products will be generated that will be excreted from the amino acid metabolism. So they are precursors for different nitrogen containing compounds like heme is one good example where nitrogen molecule is there. And nucleotides, you can see in all nucleotides will have nitrogen molecules. Different types of amines, those act as very important role in biological system. Amines are also containing nitrogen, so amino acids can act as precursors. Nucleotide coenzymes, so for example, NAD plus molecule, if you take NAD plus molecules play a very vital role in metabolism for accepting and donating its electrons. So NAD is also one nitrogen containing molecule. This can be synthesized from the protein or amino acid catabolism. And glutathione, we have discussed about this glutathione in pentose phosphate pathway. These glutathione act as a free radical scavengers. So it is very important, when metabolism occurs in cells, continuously free radicals, negative molecules, hydroxyl or hydrogen negative molecules will be hydroxyl peroxide molecules will be formed. So those are free radicals. So they have very reactive tendency. So they will interact different molecules. So to and interact and cause the destruction of those molecules. Okay, to prevent such kind of accidental damage by the free radicals, glutathione is one of the important compounds in biological system that scavenge, that remove the free radicals from the system. So glutathione is also one of the precursors, comes from the amino acids. Okay, so and also amino acids are precursors for different types of keto acids. So here pyruvate, you can get pyruvate, oxaloacetate, alpha ketoglutarate. So they are all some way or other way they are entered into Krebs cycle. Okay, all these molecules, you can see all these molecules are precursors or seeding molecules for the Krebs cycles. So from these keto acids, you can get different types of macromolecules, different types of molecules like fatty acid and ketone bodies. Yesterday we have discussed. Okay, so here you can see the intracellular digestion, intracellular catabolism of amino acids. So intracellular molecules, proteins will be there, different types of proteins will be there. These proteins have lifespan, half-life. Okay, so once these proteins are their function is over, they have to take in further degradation. So once they degrade, protein molecule degrade into amino acids. Okay, so amino acid pool will be rise here. So not only that intracellular molecule, we take diet in diet, different types of proteins will be there. So different types of protein content will be there. So that from that protein diet, after digestion, they become to amino acids by the action of proteases. So protein molecules digest and then intracellular amino acid pool will rise. Okay, so this amino acid pool will be taken in different ways for the excretion by ammonia and ammonium compound or maybe utilized for different types of synthesis of nitrogen containing biological molecule. And keto acids. Keto acids also generate from these amino acids, and these keto acids further metabolize, further oxidized by citric acid cycle. So you have seen different types of intermediate molecule in the citric acid cycle, which basically from keto acids input. Okay, so I don't want to take much on this, classification, amino acid classification. You might know that amino acids are classified into essential amino acid, non-essential amino acids. So essential amino acid, basically, the supplement is required, dietary supplement is required. So those are called as essential amino acids, and those are play a very important role that particularly. So here you can see essential amino acid and non-essential amino acids. So arginine, histidine, isoleucine, leucine, lysine, methionine, threonine, tryptophan, valine. They are belongs to essential and required from the dietary sources. Arginine is in young, growing animal does not but not in adults. Okay. We will talk about how amino acid degrade. Okay, so instead of talking about amino acid classification, different classification, people use polar, non-polar, side chain amino acid, acidic, basic amino acid, all those things. So those are all basic things. So we'll avoid that here. So how amino acid get degrade and get deaminated. Okay, so amino acid degradation first starts with deamination reaction. Okay, breakdown of amino acid first start with deamination. So here you can see this is amino acid. Amino acid when it is deaminated. Okay, so it become other amino acid. Okay, so amino transfer is here. Alpha ketoglutarate is the one. Okay, so that remove the amino group. Okay, and it forms as a keto acid. Okay, and glutarate molecule is further further goes down to aspartate. So here oxaloacetate is the input molecule, and alpha ketoglutarate form here. This alpha ketoglutarate may again join here as a cycle form here, and from glutamate aspartate form, and from aspartate you can see via urea cycle this aspartate will metabolize, and then amino groups may be excreted as urea from the higher animals, and fumarate is formed. So fumarate again, intermediate molecule for the TCA cycle. So, and from glutamate, nitrogen containing compounds may also, different biological nitrogen containing compounds may also generate here. Okay, so overall transaminase is the one first reaction that transfer the amino group from the amino acid, and this reaction you can see occur in two steps. Amino group plus alpha ketoglutarate, keto acid will be joined, keto acid enter into TCA cycle, and glutamate interact with oxaloacetate in the presence of oxaloacetate. You will get alpha ketoglutarate and aspartate. Aspartate again become to fumarate. Upon using fumarate, it provides nitrogen containing waste product ammonia, urea. So that that will be excreted from the system in higher vertebrate animals. So here, glutamate converting to alpha ketoglutarate is a kind of oxidative deamination reaction. So here, high energy compound will, NADPH plus H molecule will generate. So glutamate in the presence of alpha ketoglutarate, in the presence of NADPH, it will convert into alpha ketoglutarate and as well as aspartate. Okay, this reaction is oxidative deamination reaction that generates NADPH plus hydrogen molecule. So it has very important biological role in catabolic reactions. Okay, so amino acid oxidase, you can see amino acid plus FAD, you can get FADH2 molecule, ammonia molecule, which can be used for other nitrogen-containing biological molecule synthesis are excreted as urea in the higher animals. Okay, so FAD again acts as, it can be oxidize and reduction can be takes place to generate H2O2, hydrogen peroxide. Hydrogen peroxide further metabolized into mm, hydroxide molecule, oxygen species, reactive oxygen species. So those are called as make what free radicals, reactive oxygen species. These reactive oxygen species can be controlled or regulated or scavenged or removed by the glutathione, which you have seen in previous classes. Glutathione is act as a removal of oxygen free radicals, oxygen reactive molecules. Okay, any doubts so far?
No, sir.
All right. So this transamination reaction you can see here, amino acid interact with the enzyme, transaminase enzyme, and alpha keto acid will be generated, and the enzyme hold the amino group here, and then this enzyme which is having amino group interact with the alpha keto glutarate. Okay, so this amino group from alpha enzyme group enzyme molecule will transfer to alpha ketoglutarate and it will become glutamate molecule. Okay, then glutamate molecule again oxidative gamination. Okay, that will give all aspartate molecule. Okay, so here this transamination reaction require a cofactor which is called as pyridoxine, which is known as B6 vitamin. So pyridoxine in here in this enzyme present as a pyridoxal 5-phosphate, and pyridoxal 5-phosphate when once the amino group is transferred from amino acid to the enzyme, the pyridoxal 5-phosphate hold the amino group and it becomes pyridoxamine 5-phosphate. Okay, so from this pyridoxamine 5-phosphate, the enzyme holds amino group and it will interact when it interacts with alpha ketoglutarate and it will donate amino group to alpha ketoglutarate and make it glutamate molecule. Okay, so this is the reaction. So you can see the mechanism here. And this is about pyridoxal 5-phosphate. So amino transaminase reaction occur via ping-pong mechanism, whose have 3 steps each. Okay, so here we have 2 steps. One is removal of amino group from the amino acid, and it holds, and then it will donate to amino groups to another substrate here. Okay, so the removal of amino group is 3-step reaction. The same way the donating the amino group to substrate is also 3 steps involved. So these reactions, ping pong chemical reactions, you might see. So you might familiar with that. So like ping pong reaction, this amino transferase will occur here. Okay. So first, how it will take. So first, the amino group nucleophilic amino acids nucleophilic amino group attacks the enzyme LPP. This is pyridoxal 5-phosphate skip space. The skip space formation lysine is the one which is present in enzyme, so that forms as a skip space here. Skip space carbon atom in a transamination reaction to form unneated pyridoxal 5-phosphate skip space. So aldehyde will form with concomitant release of enzyme lysine amino group. The lysine is then free to act general base at the active site. So the amino acid PLP skip space tautomerizes to an alpha keto acid, which is called as pyridoxamine 5-phosphate skip space, to ketamine, which is known as ketamine. By active site, lysine catalyzes removal of amino acid alpha hydro and protonated PLP atom at 4th carbon via resonance stabilization carbanion intermediate. Okay, this resonance stabilization facilitates the cleavage of the carbon alpha carbon hydrogen bond. Okay, so the keto acid PNP skip space is hydrolyzed to PNP and a keto acid will be released. So this keto acid is now which is lose the amino group. So this amino group will be present in the engine on engine structure as pyroxamine biphosphate. So now the pyroxamine biphosphate give back to another keto acid, donate its amino group. Okay. So to complete the amino transferase catalysis reaction, the coenzyme must convert from PMP to PLP skip phase. This involves same three steps reaction. PMP reacts with keto acid to form as skip phase. Alpha keto acid PMP skip phase tautomerizes to form as amino acid PLP skip phase. So E amino group of the active site lysine residue attacks the amino group PLP skip phase in transmission reaction, generate the active PLP skip phase and release newly formed amino acid. Here, glutamate is the newly formed amino acid. So here it will impact our alpha ketoglutarate and it donates it amino group, okay, by tautomerization at the active site, okay, and then it donate amino group to keto acid, and you can see newly formed another amino acid, which is known as glutamate here. So this is a mechanism of how first reaction will take place, how amino group interact with the enzyme. Enzyme lysine molecule is the skip phase, and PLP and PLP amide group will form here, and this taking on the tautomerization that will sink electrons here, so that will take the carbon alpha carbon and hydrogen molecule bond here, so that then that enzyme will hold amino group here. So then the enzyme will release keto acid here after hydrolysis after that, and then PNP amino group will have amino group is present on the enzyme here, and then it will interact with the other way to donate PNP enzyme and keto acid, and again convert back to PNP enzyme with hydroxyl biphosphate, and then glutamate, another new amino acid formation we can see here. Okay. This you can see resin stabilization. So resin stabilizations, in this reaction, transamination, decarboxylation, removal of our group. Here, it may be amino group here. R group means. So, degradative facts of glutamate, regeneration of alpha ketoglutarate. So, once glutamate is formed, so this glutamate again oxidative deamination, through oxidative deamination, it will regenerate alpha ketoglutarate. Okay, NADH plus hydrogen molecule, NADPH plus hydrogen molecule formed here. So here you can see amino acid alpha ketoglutarate transaminase reaction. It interacts with in the presence of alpha ketoglutarate. So it forms as a alpha keto acid because of transaminase reaction in presence of another keto acid. So new amino acid form, that is glutamate. Okay. So this glutamate can act as per synthesis of nitrogen containing compounds or ammonium ions, and oxidative deamination, it may regenerate alpha ketoglutarate in the presence of oxaloacetate to aspartate. So this aspartate, upon hydrolysis taking different steps, it loses fumarate molecule via urea cycle, and urea is produced, and this can be excreted from the system. So this is the oxidative deamination fate of the glutamate. Glutamate plus oxaloacetate, it will give alpha ketoglutarate and then aspartate. So NADPH is formed here. Glutamate, immunoregulate glutamate. So here removal of hydrogen molecules here. So you will get NADPH molecule, and alpha immunoglutarate, and upon hydrolysis it will give ammonium ion molecule, ammonium ions, and then alpha ketoglutarate will generate here. So this reaction can be regulated by ADP molecules, high amount of ADP molecules, NADPH molecules, and it can be inhibited or stopped by high concentration of GTP molecule, NADP plus molecule. So that is the starting point of amino acid deamination reactions. So if you see amino acid deamination reactions, amino acids will be break down into different precursor molecules, keto molecules, and some are keto molecules. Those molecules can be divided into different molecules. Here, amino acids are degraded to compounds that can be metabolized to carbon dioxide and water, or used to gluconeogenesis. Indeed, oxidative breakdown of amino acid typically yield 10 to 15% of the metabolic energy generated by animals. So, standard amino acids are degraded one of 7 metabolic pathways intermediates. So, here all amino acids can be separated based on their metabolism. You can see amino acids, those give pyruvate, amino acids, those give alpha ketoglutarate, and amino acids, those give succinyl CoA. Some amino acids, those give fumarate, amino acids, those give oxaloacetate, acetyl CoA, or acetoacetone. Okay, so when you see all standard amino acids, when you observe their metabolism, so amino acids can divide based on what kind of substrate end product they give. Okay, so you can see 7 metabolic intermediate molecules are generated. Approximately 7 intermediate molecules are generated from the standard amino acids deamination. Okay, so amino acids can therefore divide into 2 groups based on their catabolic activities. One is called as gluconeogenic amino acid and ketogenic amino acids. So you have seen those end products. These end products, those are enter into gluconeogenic pathway or ketogenesis pathway, ketogenic pathway. Okay, so particularly amino acids, those are divided into pyruvate, alpha ketoglutarate, succinyl CoA, fumarate, or oxaloacetate, are therefore enter into gluconeogenic pathway, either Krebs cycle or gluconeogenic pathway, to give rise to glucose molecule. Okay, to give precursors, glucose precursors. Okay, and ketogenic amino acids, which are break down to acetyl CoA or acetoacetone, thus can be converted into fatty acid or ketone bodies. So acetyl CoA and acetoacetone. So we have seen fatty acid beta oxidation. They give acetyl CoA. If you see more, the ketone bodies will be synthesized as acetone acetate molecule. So they are the precursors for the fatty acid synthesis. So ketone, fatty acid degrade into acetyl CoA. From same acetyl CoA, the fat molecule can be synthesized. Okay, so they are precursors for the fatty acids and ketone bodies. Some amino acids are precursors for both carbohydrates and ketone bodies. Some amino acids they are ketogenic as well as glucogenic molecule. Since animal lack any metabolic pathway for net conversion of acetyl CoA or acetoacetone to gluconeogenesis precursors, no net synthesis of carbohydrates is possible from the ketogenic amino acids, particularly lysine and leucine. This was we already discussed in gluconeogenic pathway. These 2 amino acids particularly give acetyl CoA and then acetoacetone. So there is no enzyme that converts glucose molecule or glucose precursors from this acetyl CoA or acetoacetone in animals. So these 2 amino acids are called as ketogenic amino acids. So now we are going to see one by one how many amino acids, what are the end products of these amino acids. Here we have discussed basic mechanism how amino acid is degraded or how amino acid can be classified based on the end product, glucosenic or ketogenic. So what are the ketogenic and ketogenic end products, acetyl CoA or acetoacetone, and what are the glucosenic end products like pyruvate and alpha ketoglutarate, succinyl CoA, fumarate or oxaloacetate. So you are going to now we are going to see the different types of amino acid deamination here. Next, so any doubt here? Next slides doesn't show much, just the pathways only. So if you have any doubt, we can revive the mechanism or we can discuss on this. Any doubt?
No, sir.
Okay, so now you can see different types of amino acids. Standard, we have 20 amino acids. Okay, so you can see different types of amino acids like threonine, glycine. From threonine you can see formation of glycine. From the formation of glycine, from glycine you can see formation of serine. So from serine, pyruvate is generated here. So threonine basically gives pyruvate. Okay, so cysteine also gives after 2 steps pyruvic acid, and tryptophan after 4 steps you can see alanine, then it will convert into pyruvate molecule, and then after that pyruvate molecule, as you seen in Krebs cycle, is the precursor for the acetyl CoA molecule. So here you can see pyruvate dehydrogenase, which remove the carbon molecule to generate acetyl CoA molecule after conjugation with the coenzyme. So glycine we have seen here. Glycine will come generate serine molecule, or glycine can be degraded into carbon and ammonia here. So glycine after conversion, say serine hydroxymethyl transferase, it can be converted into serine, another amino acid, and another pathway, glycine synthase, that can this reversible reaction from carbon dioxide ammonia, it can be synthesized as a glycine in the presence of glycine synthase, and it can be degraded into after oxidative deamination. It can be converted into carbon dioxide plus ammonia, and NADH plus molecule, hydrogen molecule will be formed here. So catabolic pathways of aspartic acid and asparagine. Asparagine, in the presence of asparaginase, it becomes to aspartate. Aspartate aminotransferase, it makes it to oxaloacetate. Oxaloacetate is one of the independent molecule for TCA cycle. So catabolic pathways of arginine, histidine, protein, and glutamic acid and glutamine. So here you can see one way or other they become different keto acids like alpha ketoglutarate. After few steps they become alpha ketoglutarate. So catabolic pathways of tryptophan, tyrosine, phenyl, and lysine. So they are become acetyl CoA or acetyl molecule or succinyl CoA. So you can see all these are keto acid. All these molecule keto acids, as well as gluconeogenesis intermediate molecules. After 1 or 2 steps, they become acetyl CoA molecule, or acetoacetate, or succinyl, or pyruvate. You can see fumarate. All these molecules both are present in ketogenic as well as gluconeogenesis pathway. So tryptophan gives some important biological molecules. So NAD precursors, okay, indole acetate, which is known as plant growth factor, serotonin, which is known as neurotransmitters. During its metabolism, during its degradation, It can give different types of nitrogen-containing biological active compounds like nicotinamide, niacin acid, NAD, NADPH is the component you can get from this, and indole acetate and serotonin. These are very good, very important molecules like the transmitter and growth factors, act as transmitter and growth factors. So here you can see phenylalanine and tyrosine, their degradation. If amino acid degrading enzymes, particularly those enzymes have problem, you can see kind of diseases here. This kind of condition, pathological conditions you can see. If phenylalanine hydroxylase is mutated, phenyl PKU, phenylalanine urea, fructonuria, I think, is PKU condition will be present, and tyrosine aminotransferase. If you see tyrosinemia, which is present in blood, tyrosine amino acid, high amount present in serum, okay, and alpha P hydroxyphenyl pyruvate dehydrogenase. If this molecule is not, this enzyme is not active, tyrosinemia, T condition will be present. And here you can see tyrosinemia 1 condition. If fumarate lacto fumarate acetoacetate is defect, so you can see tyrosinemia condition. Okay, so here it is already marked which enzyme has pathologically important in the amino acid, particularly phenylalanine and tyrosine degradation. So you might see in metabolism of this catabolism of these reactions, tetrahydro, which one is the role of tetrahydrobiopterin in phenylalanine hydroxylase reaction, which is which is the one that acts as a strong reducing agent here. NADH molecules can be formed as NAD plus molecule. Okay, so tyrosine to phenylalanine will form here, which 4, 5, 6, 7, 8 tetrahydrobiopterin molecule play an important role to synthesize in this compound phenylalanine molecule. So alternatively, phenylalanine can be degrade into phenylactine and phenylactate. So these two molecules, the end product of phenyl pyruvate accumulates in liver and blood and urine. Phenylactate and phenyl acetate and phenyl lactate also found in urine in these conditions. So all these things, basically they undergo first activate amino acid catabolism by deamination reaction, and then different molecules from these different steps they will end up into different gluconeogenesis precursors or ketogenesis pathway precursors. Okay. So catabolic pathways of 3 branched chain amino acids, particularly valine, isoleucine, leucine. So here you can see if some problem is here in this metabolism, branched chain keto acid dehydrogenase complex is the enzyme which is involved for the catabolism of these branched chains. If this enzyme is defective, you can see maple syrup urine disease. So urine will be pale red color, okay, like honey color. Okay, when you dilute honey, you will see kind of syrup, maple syrup color. In jaundice, the urine looks like yellow, whereas in maple syrup disease, so you will find in this enzyme defective, you will find maple syrup urine disease. So here amino acids, ketogenic amino acid and gluconeogenic amino acids are depicted here. So all these molecules, different molecules are metabolized via Krebs cycle, which is PCA cycle. You can see that's why Krebs cycle is the one which is called as hub for all catabolic reactions. So here you can see different diseases which are defect in amino acid catabolism. We have seen in previous slides, and here also I given in the table form. Just please take it later. So why ammonia is toxic? So ammonia, as you know that the waste product ammonium NH plus NH4 plus molecule, if it is there, it is excreted. If it is removed from the cell, so it has to excrete in the form of urea. Okay, so why it is toxic? Exactly, we don't know the reason why it is toxic, but some speculations. Ammonia posing a poisoning is accumulated by corneal edema. So if you find ammonia excretion defects, you will see swelling of the body. Okay, and this ammonium can be removed by two ways from tissues: amide to alpha glutarate to glutamate. So it can be used to aminate the alpha ketoglutarate to become glutamate acid, aminate glutamate to glutamine. Okay, so glutamine will contain two amino acid groups. So one glutamate it get aminated and then become glutamine molecule. Okay, when ammonia is high, so you can see glutamine is also, or concentration of glutamine is also high. Okay, so here alpha ketoglutarate to glutamate it will form, and again glutamate again get aminated, it will form glutamine. So the marker for this ammonia high concentration is measuring the glutamine in the serum. So if glutamine is present, that indicates ammonia is high. Okay, so glutamine can enter in neurons and also osmolar solid, leading to water uptake by these cells. So glutamine, when glutamine is high, so it will enter into cells and neurons and osmolar solids. So it absorbs the water molecule from the surroundings. So once the water molecules are absorbed from the surroundings, cell will become bulge. Okay, so certain levels only the cell can become bulge. After that, if keep on concentration is going on, it causes the destruction or causes the lysis of the cell. So that will give the problem. Or even you touch the swelling part, you will feel heavy pain. So this is why ammonium should be removed from the excreted from the system. So summary of this amino acid catabolism: amino acid interact with keto acid, it will give alpha ketoglutarate, it will give keto acid, and keto acid will further metabolize into carbon dioxide and energy products. And again, glutamate, new amino acid will form. So this glutamate, in the presence of oxidative glutamate dehydrogenase, it will convert into oxaloacetate, aspartate, and then aspartate via urea cycle. Ammonium molecule will be removed as urea from the system. Okay. Okay. Any question? Tomorrow we'll see urea cycle.
Sir, tomorrow at what time, sir?
Oh, tomorrow means tomorrow you have class. You have any free time? Do you have any free time tomorrow?
Yes, sir. First hour is free actually, 9:30 to 10:30.
First hour 9:30 to 10:30. Okay, I will try to join. Okay, be ready.
Okay, sir.
Okay?
Hello, sir.
Yes,
Sir, actually, have you received any mail from Pennsylvania University? I have
yes,
given your name for the reference, sir.
yes, I have received.
Okay, sir.
Working on that.
Sorry, sir.
I'm working on that.
Okay, sir. Okay, thank you, sir.
Right. I think you might have seen university circular in our university website regarding your arrival.
Yes, sir. We have, sir.
Okay. So they are strictly saying that they are allow you from sixth onwards. That is the meaning of that circular.
And they have mentioned some selection, no, sir. When we will be notified, actually.
That they said 10 days before that they mentioned in that circular. I think.
Yes, sir. It seems 1 week or some
Mm-hmm.
time frame they have given.
Yes.
It
Yes.
will be received from you, actually. They told from HODs like that.
Yeah, once we receive, we'll immediately display to you all our through our website. Website means our department website, and it will be sent to all your groups,
Okay, sir.
mail groups. Okay,
Sure, sir.
right. Okay,
Sir, it is just a request. Please submit, sir, your reference, a reference for that university because the deadline is coming soon. So
sure, I'll
that
do.
is
Mm,
okay, sir. Thank you, sir.
right. Okay, if you don't have, I will sign out. Some calls are coming.
Thank you, sir.
Thank you.